The cell cycle is the life cycle of the cell, from when it is first made until it divides again. And the cell cycle is made up of these phases that we go through in a specific order. So the very first and the longest phase is called interphase. Inter means between. And what it's talking about is it's between divisions. So this is what's happening when the cell is not dividing. So the cell is just doing its normal cell thing. It's just chilling. If it's a muscle, it's just being a muscle. This is when it's not. So interphase is made up of 3 parts. We have G1, S, and G2. So that entire section there is interphase. G1 is called gap 1. You can associate the G with growth, if that's easier for you to remember, because it is growing. S is synthesis, just like DNA synthesis. So this is when DNA is being replicated. And then G2 is called gap 2 or growth 2 because it is also growing. And when all of these phases are complete, the cell has gone through interphase. And if you look at the cell cycle, it looks like interphase is like 75% of its life cycle. That's not to scale. Interphase is actually going to be much larger, more like 90-95% of the life cycle. Mitosis is this M phase right here, which is when the cell's actually dividing. And it's going to go through prophase, metaphase, anaphase, and telophase. Until at the end of this, we have 2 cells that are identical, and then each one of them is going to go into their own interphase again. So why divide? Cells are going to divide to replace themselves, right? So if a cell is damaged, it can be replaced. It can repair sections, like if you get a cut on your arm. The cells that are there don't just get bigger. What happens is you create new cells to replace that damage. And also for growth and development, because cells have a maximum size. They can't get very large. So if you want to grow into a larger human being, like you do through childhood, you need more cells rather than bigger cells. When we talk about mitosis, this is the division of every single cell except for eggs and sperm. And we'll talk about eggs and sperm. This is going to be in a process called meiosis. And you'll do this more in anatomy too. But for now, mitosis is going to make 2 identical clones. So we're going to start with 1 cell, and we're going to copy it and make 2 cells, and they're going to all be clones, meaning identical. The only reason that there would be any difference is if we have a mutation error, and that's not the intent. The intent is to make them identical. So we're going to go through 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. So if you think about what these words mean, pro means before, kind of like prototype. So you would make a prototype of something before you actually just sent something off to a manufacturing line to make a bunch of them. Meta means middle, and that's going to come in handy for remembering where the chromosomes are. Ana's a little tough. This means up, from. This is association with motion. It's during the anaphase that the chromosomes are actually moving apart. And then telo means end. If you think about the word telephone, we used to have telephones that were attached to cords, which were attached to the wall, and you had to actually stand in one spot to talk on the phone. But, now we all have cell phones. But telo was because you held the phone at the end of a cord. So telophase means end. This is the last phase. And then cytokinesis is technically not a part of mitosis. It's actually its separate process. The word cyto means cell, and then kinesis means move. So this is cell movement, and this is where the cell is going to actually divide into 2 separate cells. So let's look at each of these phases. So you need to be able to describe what cells are doing in each of these phases. So in prophase, the chromosomes are condensing. I like to think about this like you're packing up a house. if I'm packing up my house because we're moving and splitting into 2 houses, right? You don't just take your belongings and throw them in the truck and take them to the new house. Instead, what you do is you condense them. You take all of your belongings and you pack them in boxes as tightly as you can so that you don't have to move as many things. Same thing's going on here. Chromatin is very much like looking like spaghetti. So what I want to do is I want to take my DNA and I want to condense it down into 46 teeny tiny packets so that I can move them easily. So we're going to condense the chromosomes. This purple thing is the nucleus, and you can see it's starting to break here. So it's just starting to break apart because if I want to move, I have to open the door. If I want to move DNA around, I need to break down the nucleus and get the doors open so I can actually take those boxes out to the moving truck. Spindle fibers are going to grow from the centrioles. So there are 2 centrioles, and these are going to migrate to the opposite sides. This is going to come up here, this one's going to come down here. The centrioles are going to determine like what's the North Pole and what's the South Pole, and they're going to tell the cell which ways it's going to divide. So the centrioles migrate, and then these spindle fibers start to form out of them. These spindle fibers are your movers. These are the guys you pay with pizza and beer to come and move your chromosomes for you. What these guys are going to do is they're going to actually physically grab the chromosomes and move them around. The second phase is called mitosis. The second phase is called metaphase. And what's going to happen here is in metaphase, all of the spindle fibers attach to the kinetochores. Remember, those were those proteins. Here's my chromosome. Those were those proteins on the sides. So the spindle fibers are going to come in and attach to those kinetochores. And what you can see is that there's a spindle fiber attached from both poles. That's important. So if we want this chromosome to split properly, I've got to have somebody tugging it this way, somebody tugging it this way. And then because it's attached to both poles, think about pulling equally from both ends. You're going to end up with the chromosomes along the middle. So the chromosomes are along the middle. And this isn't like just a line, it's more of kind of a plane. So here's the plane through the center of the cell. And so that's called the metaphase plate. And so all of the chromosomes will line up in the center of the cell, and then we're ready to move on to the next phase. In the second phase, anaphase, remember anaphase is associated with movement. What's going to happen is these spindle fibers are going to start to shorten finally. And these are just limp noodles, right? They're not very stiff. So if I grab it by the center and pull, these ends are going to kind of make these little V structures. And these little V's are a good indication that your cell is in anaphase. So this is where the centromeres divide. So we actually physically cut each chromosome down the center. And then the spindle fibers shorten, and they're going to pull the chromatids to the opposite sides. And you can see that the entire cell is kind of longer than it was. It's starting to get wider. All right, the last part we have is telophase. I like to think of telophase as the opposite of prophase. So everything that we did in prophase, we have to undo in telophase. So now in telophase, our chromosomes are going to gather at the 2 opposite sides. The chromatin is going to uncondense, so these chromosomes are going to start to get looser and looser. Because we're done, we already moved them, we don't need to move them anymore. This is basically the same as unpacking your boxes when you get to your new house. My nuclear envelope reforms, right, so I'm going to close these back into a nucleus. Once I get to my new house, I want to shut the door, we're not letting anybody in. And those spindle fibers vanish, right? Those fibers that were over here, they're gone now. These were the movers. So we send the movers home. Once we're already in our new house, we shut the door, we unpack our boxes. We don't need our movers anymore. They don't just move in with us. They actually leave. So this is technically telophase. And then cytokinesis is a distinctly separate process. Cytokinesis is going to be kind of the invagination of this membrane on both sides. What actually happens is it forms a ring of microfilaments around it and kind of zips it shut. And then eventually they will pinch off into two separate cells. So this indent right here is called a cleavage furrow, and it's going to cleave these two cells in half. A cell can do telophase and mitosis without doing cytokinesis, but usually you're going to go through all of the phases of mitosis and do cytokinesis at the end too. So, do all of your cells need to be dividing at any one time? No. All of our cells have different functions. And cells actually grow at different rates. So, we need something to tell our cells when to grow and divide and when they should not. So, think about your hair and nails and the lining of your stomach. These are all types of places on your body that are constantly growing, or your stomach is constantly getting damaged from acid, so it has to keep replacing itself. So those tissues are going to need to constantly be doing mitosis. But your heart cells and your brain cells, once you're set, your heart and your brain doesn't really divide anymore. That's why heart attacks and strokes are so damaging, because those cells don't repair themselves. So the cell cycles for those have actually been turned off. They stay in interphase. So what we have are these things called growth factors or cyclins that help tell our body when to divide and when not to divide. So a growth factor is going to say, yes, divide. So this is going to actually happens when you get a cut. Your blood platelets will come to that area and they'll release growth factors and tell your body, hey, we got to repair this cut. And once it's repaired, the body knows to stop growing because the cells touch each other. Once all of the cells are touching each other, they come in contact with each other and they say, okay, we've completely repaired this cut. We can stop dividing. That's called contact, right? Touching. Inhibition. Inhibition means stop. So when they touch, they stop growing in a normal cell. A cyclin is a protein that tells cells when it's time to divide. And think about cyclin like cycle, right? So these cyclins actually increase and decrease in a cycle. And when they increase, it divides. And when they decrease, we stop dividing. And then during our cell cycle, we have multiple checkpoints. And these are kind of like just checkpoints to see, okay, am I in the right spot to be dividing? Is my DNA copied properly? Is my cell large enough? Do I have enough organelles that I can go ahead and divide into two? And when any of these things goes wrong, maybe there's too many growth factors, or the inhibition didn't work, or the cyclins are out of control, or we get past a checkpoint that we weren't supposed to get past. If any of these regulating factors, right, these are all regulating factors of the cell cycle go wrong, we can get what's called unregulated cell growth, and that is cancer. So cancer is a really generic term for just your cells are growing, but they're not supposed to be growing. They're dividing and they're not supposed to be dividing. So there's two different types of cancer. You can go to an oncologist and you can say, oh, you've got a benign tumor. A benign tumor is, here we've got the cancerous cells are in blue here, but they're all maintained right here. So at one point, one of these cells became cancerous. It had a mutation for one of these different checkpoints, and it started dividing. And you can see that the contact has done nothing. It's just decided to grow up instead. And from that one cell, we've kind of created this tumor. Benign means it has not spread to another tissue, and it probably won't. And so for benign, that's good news. You go in, this is maybe like a polyp in the large intestine, the colon. And so what a doctor would do is you would have maybe surgery to go in and remove these. And if you watch the medical shows, they'll say, oh, I got good margins. Good margins means we think we got it all. Because if it's benign, you can remove all of these cells and only leave behind the normal cells, and you should have removed the cancer completely. A lot of the times, a surgery will be accompanied by something like radiation or chemotherapy to make sure, so that if there are any single cells left, that it doesn't come back, that they kill those cells. A much more problematic cancer, metastatic. And this is when one tumor forms. So here's the first tumor. One of these cells breaks off and somehow travels and creates another tumor. This is called a met. And this can happen by moving through the lymph system or the circulatory system. It can even happen if it doesn't travel like this, but if it just invades another tissue. And so a metastatic cancer is a more aggressive cancer. This is going to be your like stage 3, stage 4 kinds of cancers that have moved. And sometimes surgery might be okay, like if it's a large tumor, a surgeon might go in and remove it. But often what they're going to do with this is we're going to do something called chemotherapy. And chemo is a very toxic drug. And there's lots of different chemotherapy drugs. Chemo is a anti-mitosis drug. So what chemo is going to try to do is try to stop these cells from dividing, and it's going to try to poison these cells faster than it poisons your cells. So take a minute and think about what are some side effects of chemotherapy. Right, so if you've maybe had a family member or friend who went through chemotherapy, like what happened to them? Some common side effects might be things like nausea, vomiting, weight loss, hair loss, and think about those tissues. Hair, those are things that are growing constantly. Part of the reason you lose your hair during chemotherapy is because your hair is doing mitosis, and chemo is an anti-mitosis drug. The reason you get kind of nausea, vomiting, is because your stomach and intestinal linings are constantly being damaged, and so those are constantly doing mitosis. Again, chemo is an anti-mitosis drug. So the side effects of chemo come from the fact that it's actively killing cells that are actively dividing. So this is the IARC website about carcinogens. IARC is the International Agency for Research on Cancer, and they've made this little graphic here to simplify what is a carcinogen and what are some of the worst carcinogens. So a carcinogen is something that causes cancer in humans, and they're grouped into different groupings, and these are standardized throughout the world. So group 1 is something that is evidence shows that it definitely causes cancer in humans. So things like smoking cause lung cancer, UV radiation can cause skin cancer, alcohol, and then this is bacon, processed meats. So this is, going to the deli and buying deli meats, the Lunchables that you buy your kids, and yes, bacon. And so any processed meat is considered a group 1 carcinogen for specifically colon cancer. And then group 2A are things that have evidence linked, so sufficient evidence to say that it probably causes cancer. Emissions from high temp frying, so think about like frying chicken or frying turkey or frying anything in a pan that emits particles into the air. Steroids, so anabolic steroids like that you would take to enhance performance in sports. Exposures from working in hairdressers, so this is probably like long exposure from working with various chemicals. And then red meat, so red meat is going to be like cows, beef, pork, that kind of thing. And then there's other carcinogens here that are less likely to be linked. These are possibly, but we don't have any conclusive evidence, more anecdotal evidence at this point. So it gives you something to think about as far as the lifestyle choices you make and how those affect your body. So take a minute and reflect on cancer and how it can be caused.